more and more people are choosing their papers. I'm going to probably be done putting papers out by tomorrow at the latest. I thought there was one paper I saw yesterday and I should have printed it up and I didn't. It was good. So if you have already picked out your paper, you might as well come in. What the heck? Here's what you're going to do. First of all, on your homework, make sure you print out or type the title of your paper and at least the first author. Okay, that helped well. So I have all the papers and I'm going to be needing all the papers. And then, yeah, tell me the first author. Almost all, I think always, I don't even think almost always, I think always the first author listed is the person who wrote the paper. Then there's a whole bunch of other contributors, and then the last person listed is the person who's the principal investigator, which means that he or she wrote the grant to get the money to do the research and supplied the laboratory for the research. And you'll notice more and more, a lot of papers are coming from multiple institutions, so there's a lot of collaboration. And that may be explained why sometimes an experiment is done with one cell line, or one experiment uses Thapsigargin and another experiment uses tunicamycin, because there's a lot of collaboration going on. Okay, so I want you to read through the introduction. Okay, there's going to be stuff in the introduction that you don't understand because it involves proteins that we never talked about. Don't get hung up on that stuff, okay? Because what I really want you to be able to do is guess what's the overall goal, or what's the overall theme of this paper, okay? So don't get yourself bogged down too much in all of the detail, and because you're going to see too much detail in the introduction, okay? So what I really want you to do then is read the introduction to get the whole, just get the overall scheme of things. What did the authors already know? I don't care if you list all the proteins, well they knew such and such a protein was involved in this and that. Don't get caught up in the stuff you don't know, okay? So why is this important? Why would anybody, this will probably an expensive set of experiments, as we, in biochem lab, our last experiment cost us about $500. Experimentation is super expensive. Why would anybody spend the money to investigate such and such a question? So try to figure out, why is this topic important to spend a bunch of money on? Okay? And then try to figure out what is it that the authors still didn't know before they did their experiments and wanted to know. Okay? Generally, that question is asked after the experiments have been done. So there's always some hindsight in actually writing that question. So the question that they ask is not a question they formulated before they did their experiment, but after they did their experiment. And they said, well, because it always is going to match, the question's always going to match the results. What do you know? Okay, so that's number 3. Then I want you to know in number 4, so what kinds of information are you going to need to draw on from the course? So I chose papers that may have introduced 1 or 2 new proteins, but the bulk of the information is drawing on some aspect of this course. Okay? Then, so that's not going to take you a long time. So number 5 actually isn't going to take you a long time either. So you're going to go through each of the figures of your paper and write down the caption, because the caption is usually a sentence that summarizes the main discovery of that set of experiments. Okay, and then sometimes an outline or a flow of discoveries emerges as you kind of look at that list of things. Okay, and then so I sometimes think that from that list of that flow of discoveries, you can pick out a pretty good story for yourself. So here's what I want you to focus on, is to find a figure, generally not figure 1, that tells a good story and can be linked to something that we covered in class. Okay, because if you don't choose something that can be linked to what we covered in class, then you're really up a creek trying to make a connection, create new information for yourself. Okay, so I did a sample last night because I thought, in fact, I rewrote question number 5 because I thought, well, I better check to make sure this is even a reasonable expectation. So I picked a random paper from the biochem seminar a couple years ago and wrote down, so I wrote down all of the captions. There were 7 figures, so 7 sets of experiments. And I kind of looked them over and tried to decide, okay, if I were to write a paper about this, of this paper, what would I choose? Okay, so I made a choice, and I think if I were to do it again, I would shrink it even more. Okay, so I chose to look at figures 2, 3, 4, and 5. Okay, and then I'll tell you why, not the other ones. Metabolic stress, metabolic stress activates the PERK and IRE pathways. Well, we covered that in cell. I couldn't talk about it. Okay. I didn't want to cover 3, but I thought, dang, you have to cover 3. Okay. And then 4 and 5. And this is the storyline I thought I would go with. After reading all 7 figures, I decided the storyline would be as follows, and it's the ABC. That glucose deprivation in the kidneys activates PERK and IRE pathways for the unfolded protein response. The ER stress that activates these proteins causes inflammation. So I didn't want to include number 3, but I had to make a link between ER stress and inflammation, and figure 3 is the one that does it. So I wouldn't do all of figure 3, maybe 2. Okay, the PERK pathway does not cause inflammation. That would be figure 4. And the IRE pathway does cause inflammation. That would be 5. But actually rethinking about this after I finish typing it, I would do it this way. I'd skip PERK. Because what the heck? Well, I'm not gaining much by saying, oh, PERK's not involved. I would say instead, so I would have a 3-point paper. Glucose deprivation in the kidney activates PERK and IRE pathways in the unfolded protein response. And that this the ER stress that activated these proteins is also causing inflammation. And IRE, not PERK, activates, it causes inflammation. So I would make 3 points. I would choose bits and pieces of then figures 2, 3, and 5. Okay? And it wasn't until I went and listed each one and actually typed them in, actually hand wrote them and then typed them in, and I realized, oh yeah, there is a story here that I could tell. There's other stories embedded in there, like they talk about NF-kappa B, but I don't have to talk about NF-kappa B to tell a story from this paper. They talked about 6 and 7. They went back into real rats and did some experimentation. Everything else was done. Everything else was done in tissue culture. They did a transplant in figure 7. You know, they don't need to. That adds to the story, but it's not part of the story I need to tell. Okay, so all of you are just telling a piece of the story in that paper, and I would let these guys, the figure captions, kind of help you formulate what story you want to tell. Okay, it's not a cool idea to choose random things to talk about. Okay, you could, you know, you could, but it's probably not going to play out very well. Yes. Okay. So the main purpose for me for this assignment is to give you a jump start on your very last thing for this course. Okay, I don't want you to wait until the very last minute, which people have done, but not terribly successful. Okay, so this will give you a jump start. Okay, and I will, like I said, I'll try to get 1 or 2 more papers on. I don't think, I'm probably going to be looking more at some things I've archived over the years. I'm choosing papers from 2012 and more correct. And sometimes in some of these long-standing pathways, it's kind of hard to find current stuff unless they're unorthodox. Oh, again, and here's what else we can do with this pathway, unless there's some unorthodox thing. Okay, does that sound okay? I would do this, yeah, Kelly.
Oh, I had a question on how you want this formatted. Like,
Oh, just the similarity. This is actually, for number 5, this is not part of your, I'm not gonna consider this part of the writing thing. For number 5, I would do this, just really short. Okay, here's what the figure said, and here's gonna be my storyline. And wouldn't you know, in creating your storyline, wow, that's great organization. Because I've noticed that most people your age anymore don't write from an outline, because your writing would come out much, much differently if you did. But this forces a storyline or an outline, and so you try to use that.
For the rest of them though, is it okay if they're the number and then complete sentences, or would you like them in paragraph form, or how would you like
I
those
would definitely like complete sentences.
Yes.
Since you are now close to
I
being
was saying whether
seniors
you
and
want
this
a
is
bullet
not a
point.
foreign language, so for most of us. So yeah, complete sentences, please. Because you're still communicating to someone.
It was more the question between just sentences with the numbers or paragraph
I
form.
really at this point don't care.
Okay.
Okay? I really at this point don't care. This is not a composition thing.
Okay.
I'm not grading it for composition. I'm just kind of looking, I'm going to probably have this printed up as I'm reading through your paper. So I can maybe follow the outline. And I guess it's mostly to get you guys a jump start and form, how you think. Yeah, Amanda.
Have you graded our last homework assignment?
No, that's going to start tonight, and it's going to go through the weekend,
Okay.
the long week. No, this was letter of recommendation weekend.
Yeah.
So I'll start tonight, and I will give them back to you on Monday. And my goal this time, sort of like last time, is to say, do this in your final paper, change this. Don't do this for your final paper. So it's really going to be thinking ahead at your final paper. What interpretation ways are helpful?
When would you like this due? When would you like this one due?
I'm going to have this, you know what, if it was me, I would do this during Thanksgiving break and just get it off the table. I'm going to try to, before Thanksgiving break, have the assignment uploaded, but it's going to be due Wednesday, a week from Wednesday.
Okay.
But I would not wait that long. If it was me, don't wait that long. Right. Oh. And if anybody wants to help,
Oh, so
I think if I did like 4 or 5 a day, I would be, it would just go through my life. Okay. But I'm taking Claire swimming on Wednesday. She has no fear of water. Okay. Any other questions about either this assignment or the final paper? I will hand out probably next Monday my major pet peeves you want to avoid, because the best thing to do for your final paper is round nose as much as you can. Okay? Just round nosing. Okay. So today I'm going to want to introduce, or now getting back to apoptosis, I want to introduce to you the players. And I always think, and it always entertains me to look at who the big players are in apoptosis, because it sounds like a bunch of kids with little toy soldiers setting up a battle. And it's always a battle, and it's a matter of who wins whether or not a cell commits apoptosis. And the battle, as I said earlier, is taking place at the mitochondria. So you have the battle of mitochondria. Okay? Okay, so the players are really components of the intrinsic pathway. So I think sometimes, we could do game playing like they do in history. Just assign everybody a different protein name. You can play that proteins. Okay, so even though this is definitely in the intrinsic pathway, okay, many times the extrinsic pathway also gets involved in a fight at the mitochondria, okay? And so I'm going to say often the extrinsic pathway gets involved. Okay, so this is predominantly applying to almost all forms of apoptosis. Okay, so I'm going to start at the very bottom. So if you're going to if you're anal like me, start at the we're going to start at the right there. Okay, we're going to kind of start in the middle of the page and look at what has to happen in order for apoptosis to happen. Okay, so and we're in the mitochondria. I'm going to put my picture all the way at the bottom though. Okay. So here is the electron transport chain. There are 4 complexes in the electron transport chain. And yes, they are involved in apoptosis. In fact, an apoptotic assay is the loss of electron transport system activity in the cell. Okay? Electrons are transferred from complex to complex by a sort of mobile carriers. Okay, ubiquinone is one of the carriers. It can float back and forth between a couple of complexes. The protein cytochrome C is another carrier that transports electrons from complex 3 to complex 4. It's the only protein of the electron transport chain that is not an integral membrane protein. Everybody else is an integral membrane protein. Okay, so it's just loosely connected, and it has to be because it's bouncing back and forth between complex 3, complex 4, complex 3, complex 4. Okay? So this is the inner membrane, by the way. Remember. Okay, the inner membrane of the mitochondrial complex. And then there's an outer membrane of the mitochondrial complex, or of the mitochondrion. And sometimes I don't even know why there is a membrane there, because it doesn't do much. Well, maybe all it does is hold the protons from the electron transport chain. Okay. When a cell is given a signal to commit apoptosis, usually this is an intrinsic signal, okay? DNA damage, ER stress, something or other. Cytochrome C is released. Oh, and something else, okay? And I'm gonna wait for the something else until we get down to this story of look at the whole pathway. This is still open. Okay, well, once that happens, the electron transport system is, or chain is done. It can no longer create a proton pump to power ATP synthesis. So this is sort of like why do that? Okay, but it's the cytochrome C is responsible for activating a caspase. Okay, and this is kind of right now oversimplification. We'll get there. Okay. Okay, and the caspase will digest, let's just say, key proteins. Okay, so caspases used to be called, they went through a variety of names. I remember when they were discovered, and then there was this variety of different names that they went through, and finally somebody settled on caspase. And it makes good sense, okay? There's always in the target protein, in other words, caspases substrates, okay, the things that it's going to cut, an aspartic acid. And I put these up. Different caspases recognize different sequences in the protein. Crap, that's gonna be hard to put that together again. Okay. So, for example, and tell me what you think, what's common about these? This is a single letter abbreviation. Okay, so caspases recognize different sequences in the protein. And each caspase recognizes a different kind of a sequence. Okay, so here, and after they find this sequence, they cut the protein right after that D. What do you suppose D is? It's aspartic acid. Okay, so aspartic acid, all caspases cut proteins after an aspartic acid residue in which is really embedded in a particular sequence. Okay, so DEVD is I think is a sequence that almost all caspases recognize. Some better than others. So aspartic acid, glutamic acid, valine, aspartic acid, and then the caspase cuts after that. Okay, so it just fragments the protein. It doesn't chew the protein up into individual amino acids. It just fragments the protein. So the protein loses its function. And then other probably proteases can knock it out a little bit farther. Okay, so that's where the ASP comes from. Okay, every caspase cuts its target protein after an aspartic acid. Okay, the C stands for cysteine. Okay, because that's in the active site of the caspase. don't know the mechanism of cutting. Okay, but it's certainly not like a searing. Okay, so the main war that's being waged about, well, will I commit apoptosis, will I not commit apoptosis, is happening at the release of cytochrome C. The release of cytochrome C is highly, highly regulated. Okay, and it's regulated this way. Okay, yeah, hang on to your hats. Here come the protein names, but I think this will help you get it all out on one piece of paper. Proapoptotic proteins are going to stimulate the release of cytochrome C by one mechanism or another. And you'll need to keep track of those. There's only two mechanisms. There's only two ways that this can be proapoptotic. Okay? Okay, there are other proteins that are proapoptotic, but they're not involved in the war of should we get cytochrome C to be released or not. Okay. Okay, so 3-protein FASTAC and Bach. Okay. I don't know about Bach. I have never seen a paper or any information about Bach. So I listed it and we'll put it in parentheses. You'll hear about Bachs and Bach. And Bach. Okay, that's a B-A-K. Okay. Then there is a list of proteins, and hopefully I will describe these different classes of proteins for you. Okay, called BH3 only proteins, and I'll describe what that means to be a BH3 only proteins. This was the subject of our biochem lab, our seminar last year, because there are a lot of peptidomimetics, proteins that you can design to bind to these. Okay, they are not in any order: BID, BAD, BIM, HUMA, NOSA. I just thought, so if I'm going to make Alec McIntosh call me something, what do you HUMA something? EMF, Kirk, and Zick. Okay. So these guys are going to fight for the release of cytochrome C. Okay, and then there's a group of proteins that are going to block the release of cytochrome C. Okay, and these proteins largely are present in many cells. So you know, who wins depends on who's more active at the moment. Okay, so this would be anti-apoptotic proteins. Okay, and they are CO2, BCLW, BCLXL, MCL, and A1. Okay, so if cytochrome C gets released, the cell may be on a path towards apoptosis. Because there's other anti-apoptotic proteins that can be acting after the release of cytochrome C. If cytochrome C release is blocked then the cell's not gonna commit apoptosis.
So that's like a required step.
Huh?
The release of cytochrome C is a required step.
For the, definitely for the intrinsic pathway. Yeah, I can't think of a pathway. There's probably some cytochrome C independent pathways that might involve the ER. But for almost all intrinsic apoptosis involves cytochrome C release. The extrinsic pathway doesn't have to involve cytochrome C, but almost always it does. Okay, so I wanted to look at these classes. By the way, all of the proteins that I listed, except for the casphases, okay, the BCL, all of the BCL2s, the Baxback and Bach, and all of these pH3 only proteins belong to a class of proteins called the BCL2 family of proteins. And BCL2 was the very first one of these proteins to be discovered. And then we realized, oh my gosh, there's tons of proteins that look like this. So let's take a look before we get into the exact roles of the anti-apoptotic proteins, the Baxback, and the pH3 only proteins. Okay. Start out with the BCL2, because they were the very first proteins discovered. Okay, so this is the anti-apoptotic BCLs. Okay? Okay, so there's 4 domains, and the domains are there to interact with one another. Okay, so that's what they're doing. Okay, and they have them in order. A domain called BH4, one called BH3, one called BH2, and one called BH1. Okay, and again, these are to allow the BCL family members of proteins to interact with one another. So that's the function of those domains. All of the BCL2 anti-apoptotic proteins have a transmembrane domain. So TM is transmembrane. But the transmembrane domain doesn't function like a signal sequence thing. It allows the protein to sometimes insert itself into the a membrane and sometimes come out. So it's not constitutively in a membrane like a protein that was made on the ER. Okay, so this allows